Thank you so much for joining us today. This webinar has been, put out on a very short notice, and it's been a this thing to for everyone to join us today. Thank you so much for joining us. Today's webinar is very special to me, for Mr. Dr. Aditya Patil also. It's decoding the path of research in homeopathy. We're going to have the introduction of what research is, what goes under the process of a research, from the initial data collection to the end result, and what the first thing is how to pursue something like this, and especially in a place where homeopathy. Yesterday there was a webinar, and I was telling about how evidence is very important in any aspect, in any field, in any doings. Then evidence is very important at this age, day and age, because everything needs a strong base, and that strong base comes through research and development and publications of saying that yes, we have something very true. And homeopathy has been criticized and been called pseudoscience for many years now, and the we're trying to improve. We're going along with technology. We're trying to improve the research and making it stronger day by day. And I'm very happy to have a colleague, a friend, a guide with me, Dr. Aditya Patil, who has been with me since my college days, who's been guiding me through the same, and giving me knowledge about what research in homeopathy is and what the whole process is. So this is brought to you for everybody. Just a small step. It's not the whole thing. It's just a small snippet or a glance of what research is. So I'm happy to bring this to everybody else. Yeah, I'll give the mic over to Dr. Aditya Patil, and you can take over with the slides also.
Okay, thank you so much, Rashmika. Before proceeding further for the presentation, I would like to thank Bahola group of organizations who has given me a platform for deliberating a session on today's, specifically on the topic of R&D section of the homeopathic field. Okay, so in the process, we had a lot of discussion among the titles, what to be brought up in front of the end users, the students, the practitioners. And this is what we have been came up. The title itself explains that decoding the path of research in homeopathy. Okay, so there has been a lot of questions. There has been a lot of skeptics all over the globe who are trying to understand what exactly is this field about, and how are this people working in this field are trying to understand data, and how are they replicating it into the scientific basis. Okay. So before going ahead, I would like to give a tribute to Dr. Master Samuel Hahnemann. Okay, so because the empirical source and one of the strong minds, which still holds a lot of not only the monuments but also a lot of research data previously in the empirical times. Okay, so let me give me a brief intro that the format of presentation. Okay, so this is how it is going to be, and it would be very easier for all of you to understand. So this is an attempt. Okay, so initially we would be understanding the introduction about homeopathy. Okay, what homeopathy is and what all were the publication data and how it has impacted over the globe. And second, we will go with the impact of homeopathy over continents. Third, we will have the basic research questions on homeopathy. Okay. Then we will have a database and literature review over the slides. What is EBM? Okay, evidence-based medicine criteria in understanding this. Then what are the fundamental research questions and the evidence? Over it, we will be having the preclinical research questions and evidence, case studies research questions and evidence, clinical trial research questions and evidence, systematic review research questions and evidence. Finally, we will be having an example of drug profile comparing with the Materia Medica and the research outcomes, and then ending up with the presentations with the publications. Okay, so whenever we are speaking about the introduction of homeopathy, even today itself, there are a lot of questions raised that whether this science has any scientific base, okay, and or whether this science is believed in the theory of wave or in the quantum mechanics. Okay, so quantum field theory. So that is currently a booming and understanding the streaming of how these medicines can be effective in so much of serial dilutions. Okay, so let me give you a brief. In previous past history, that 1796 was the era where Samuel Hahnemann has been experimenting with the cinchona officinalis, and the crude bark was taken into as a way of as a method of proving, and the whole picture was then brought up into the publication as an essay on a new principle for asserting the curative power of drugs. Okay, so 1796 was the first paper of and the discovery of the homeopathy, as you can quote it up. But later on, when we try to understand, as per the years were increasing, as per the time circumstances, uh, the medicines which were showing the effectivity, or there might be chances that the trials which were running were having a lot of selection bias process or the allocation process. Okay, so in 1830, again, the belladonna, it was claimed by that it has been an effective in the scarlet fever. Okay, but if you can go with this publication, okay, there are two links below it from 2006 and 2009 from the Journal of Royal Social Medicine. Okay, you will get to know that the trials, what were the lacunas in the trials. Okay, similarly, when we are going ahead in the 1835, there was a paper explaining about the inventing the randomized control trial and the Nuremberg salt test of the 1835. It is there on the Google. You can just go it and search it out. Okay, so basically the question arises that how these medicines can be brought up into the trial and can be tested with a control so that it can be differentiated from having therapeutic efficacy. Okay, and another going ahead with this timeline, almost from the year of 1953 to 2001, this was the year which had a lot of impact on the scientificity of homeopathy. Okay, so initially it was the empirical source, it was a thought process, it was a descriptive data which was in the market scenario or in the global at that time period. But when it get raised over the time, there were a lot of questions coming up that how can this be so effective, and is there any evidence, because the dilutions were so high that the traceable materials at that time and at that time of thinking was a little bit of more about the questions. Okay, so there is one publication of the Trials of Homeopathy, a critical historical account of the origin, structure, and development of Hahnemann's scientific therapeutics, and two systematic reviews of the homeopathic trials. Okay, so you can go with this publication, and there you will be able to analyze that how the data of these the empirical sources were being put forth and how they were trying to be researched at that time period. Okay, so the era from 1796 to 2001, it was a bit of challenging on the basis of scientifics. Okay, so the people from the same community had a lot of belief. So there was a conflict between the knowledge as one mind of the scientific researchers and another mind of you can say a more of a belief apart, or those of the religious followers of the homeopathic field. But in 2005, we had an alarming sign by the Lancet. Okay, now even today itself, in the epidemic of the corona, you can even have a lot of research papers, clinical features of COVID has been published into the Lancet, and those papers have been cited and even have been used as a database in even in this trials also, which are ongoing in today. So at that time, these four research papers had a lot of impact over globe. Number one was, are the clinical effects of homeopathy placebo effects, the comparative study of placebo controlled trials of homeopathy and allopathy. It had a lot of impact. Okay, then homeopathy and the growth of truth. Third, the end of the homeopathy. And fourth was the critic slam draft of WHO reports on homeopathy. Okay, so at that time, initially, there has been a lot of media influence and a lot of researchers who are quite shattered that how the studies were conducted. Okay, the PAK program was run up, and how did they came up with the conclusion that it is a placebo effect, or whether it is do have any medicinal effect or not. Okay, so further at that time period, the researchers got more interested in understanding the meta-analysis studies and in understanding the clinical trial studies, which are currently uh, on today's basis. We had a lot of evidence on that. Okay, so as we go ahead, we will have a lot of brief scenarios about it. Before going ahead, as I have explained that, first we must know what is the impact of homeopathy over the continents. Okay, so this is one of the draft of the WHO from 2019 from the traditional complementary medicine, the apex body from India, that is the Central Council of Research in Homeopathy, and the other three bodies for the Ayurveda, Siddha, and Unani, and Yoga and Naturopathy. They had this report drafted. So you can see clearly that in 1999, the members of states, means the people who were regaining as an advisory committee, it was very less. It was about 19. But as the era just went off, as the researchers move ahead, we had 58 and then we have 75. Okay, so from 2018 data, which is the latest one. Then the continental impact, you can see the African regions. We had a lot of member of state of 40, 47 out of which 29. And coming towards in India with the Southeast Asian regions, we had the seven. Okay, the Western Pacific regions had the nine. The region of the Americans were about the nine, and the Eastern Mediterranean regions have the 10, with the European regions coming with the 11. Okay, so there's a reference link below the slide, so you can just take it down, or you can just go on Google and you can just check it out. Okay, so again, the market size of these complementary medicines, you need to understand that why are these medicines so important in the markets, okay, and what impact can they give you in the market, okay. So as you can see, this is again a new draft of forecasting the data of about $1 billion investment would be ahead over the time from, and it has been estimated till 2027 that it would get a lot of height, okay. Now, the continental review also gives you, this is a pictorial I thought of explaining more about something of diagrammatic, where the R&D is established about the homeopathy. Okay, so we can see the continent as explained in the previous slides. There's the Americas there, you can see South America, you can see the Africans, you can see in the Europes, in the Southeast, and initially the startup phase of the research, you can also see in there's Australia, we have over the Americans over that. Okay, so traditional complementary medicines overview. Now, you must also know that the population, what population is interested, which population is interested in accepting this TCM therapies. Okay, so populations in developing countries using TCM for primary health care, we have number one with the Ethiopia, Benin, then India coming over it, Rwanda, Tanzania, and Uganda. Okay, and population in developed countries who have used the complementary and alternative medicines at least once, we have Germany, Canada, France, Australia, USA. So these are the percentage as per the market and as per the forecasting data. Okay, now before going to further slides, why are these questions so important? So the previous four slides, if you see, they were highlighting the market value, number one. Number two, the population based faith in using the TCM. Okay, and number three, the R&D sections. Okay, now imagine if there is an impact of any research paper which is claiming that the science, which is claiming on the science and its efficacy. So definitely there will be a lot of impact in this global scenario and even in this proceedings of the meetings and in the advisory committees. Getting it? So we when we are working in the field of research, we definitely need to see all those things, that which research paper can have a lot of impact, because in 2005 literally means there were a lot of academic chaotic situations all over the globe when homeopathy was slammed with a lot of meta-analysis data with the clinical trial pool. And further, there has been a little bit of increase in the picture. Also, there were a lot of bills and a lot of reports. So as we go ahead, we would be able to understand those reports also. Uh, this scenario, if you want to change it, so first you need to understand what are basic questions you need to approach when you're working in the field of homeopathy. Okay, now these questions have been answered in the era of 20th century also with the researchers. Even many of them have even replicated the data of these questions into various models. There has been review models also. So currently we have a lot of data pool, but many of them are unaware about what should be the questions when you are touching in the field of homeopathy. Okay, so the first question which comes to mind of everyone, that is it homeopathic medicinal effect or a placebo effect? The question is relevant on the basis of the solvent which is present as one of the most component in the preparation of the medicines and in the manufacturing unit. Okay, then second comes at how to differentiate homeopathic medicines among themselves. Okay, you have Arnica 6C, Arnica 30C, Arnica 200C. So what is the way? What are the techniques of differentiating this medicines? Okay, the potencies. Okay, then is it potentization or a solvent which needs to be questioned? So this is again an important question, that whether the solvent as a base is making the changes over the time in giving the therapeutic effect, or whether it is the potentization, the process which is giving therapeutic effect. Okay, then how do these medicines work in such high dilutions where the relation of quantum dots are challenged with the therapeutic effects? Okay, so now when there has been a lot of publications on the nanoparticles, okay, and a lot of challenges in accepting those theories also. In 2010, means the IIT Bombay group has been worked out in identifying those particles, and the same experiment was run up by the European organizations, and there has been a lot of a bit of replications has been messed up. Okay, but the review has been written into the Thiam and the British Journal of Homeopathy about the presence of quantum dots. So if it is present, then how are you able to understand the properties of those quantum dots in such a minute doses? And is it a quantum theory which can answer all the questions of homeopathy, or whether it is whether it's an experiment, or whether there is any physical entity which is going to explain the pharmacodynamics? Okay, okay, so yeah, and another question is, are the cardinal principles based on the knowledge, research, perspectives, or belief? That is another question, because uh, the seven cardinal principles of homeopathy, if you try to understand, so those are religiously followed. Okay, I'll just. Are you able to hear me?
Uh,
Hello.
yes, Aditya, we can hear you. Everyone in the chat, please refresh your browser so that you can, start hearing better.
Yeah, yeah, yeah.
Continue, Aditya. Yeah, sorry.
Yeah, let me know if anything is going around the way. Let me know.
No,
Okay.
no, no. Don't worry. Don't worry. I'm listening.
Okay, okay. So the upcoming question is about the cardinal principles based on knowledge or research perspectives of the belief. Okay. There are seven cardinal principles as we have been learning from the academics, but it's a high time that now we need to do the research on those seven cardinal principles out there. Okay. There are evidences for the law of similars also in ahead, as you will be getting a detail about it. How the research has been going up on this particular cardinal principles. Okay. Then comes up the question of rubric gradation and its validation. As the data is more of descriptive when you try to understand the materia medica, but in the repertorial process, the sequence of getting true positive and true negative data is a little bit of challenging. So you need to know the approaches, what can be used in understanding those gradations. Okay. And this is something which I would like to highlight in the last question is, are the drug development phase sufficient to understand the pharmacology of dilutions? Okay, because what have we be answering the questions are, particularly with the pharmaceuticals are, is the homeopathic pathogenetic trials, the drug proving, how the way it is conducted, and definitely when it comes towards efficacy. So the data, how it has been acquired, and how more it can be uh, get scrutinized, or get a scientific base on the monograph. Okay, so that also need to be answered over the time. Okay, so these are the basic questions. There are a lot of questions ahead also, but this word I personally thought that which might have a little bit of eye opener for today's session. And if we are able to understand these questions on the basis of R&D, then definitely the scenarios of the previously publications over the time has to be changed, and there can be a lot of reviews coming out with the positive recaps of homeopathy. Okay, yeah. So we will go ahead. Database and literature review. So this pictorial diagram just highlights that how you have to gather the data. Like for example, the background information and expert domain is the most weakest data. Okay, so whenever you're having a discussion with your colleagues or with your companions, you need to understand on what publication are you speaking, and whether that publication had any impact or are cited in a very Scopus or any JCR report journals. Okay, because there are a lot of questions arising that about the replication of the studies, when some of the studies are showing the positive effect, so it becomes a lot of a bit of debatable part. Okay, because it's not getting replicated again when you're performing the experiment. So we also need to understand whether the data was published in with a lot of authenticity or not, and that data had any follow-up of any guidelines. Okay, so over the time, over the slides, we'll be learning with this. So case report and case series are the weakest evidence. Coming up ahead are the case control studies. Then we have the cohort studies, we have the RCTs, and after the RCTs, there is a critical appraisal topics, which gives us a review about the systematics, and then finally it's the meta-analysis. So basically, this evidence of hierarchy of the database is usually used in the modern medicine and in the concept of R&D. Okay, the highest priority is given for the meta-analysis because it has all. Okay, so just one second. Yeah, so this meta-analysis are having a lot of you can say impact because all the clinical trials, specifically of the randomized controlled trials, have been included in this meta-analysis and understanding the effect size in explaining it. Okay. As you can see here, the textbooks, access medicines, and clinical keys are the weakest data sources. Midline, PubMed, PsychoInfo, Scopus, Sinhal, Trip, and these are the databases where you will have all the publications of the particular topic. Then the consultancy, and then the Cochrane database is one of the most major impacted. Okay, so over the time, how to acquire this data? Okay, now we are be understanding the questions to be answered. Okay, so if you want to answer those previous questions, we need to understand which how can we extract the data from the publications. Okay, so this slide explains you about some keywords that you need to be putting up into the search engines. Okay, like homeopathy with the two words, one is with the Latin and one is with this then in vitro studies, in vivo studies, case studies, randomized control, meta-analysis. Okay, there are three dialog box in front of you all. The first dialog box explains us about the databases usually used in segregating the data. The number one is the PubMed, the Google Scholar, we have the Web of Science, Science Direct, Sinal, Scopus. So the previous slides had a lot of references. All the references were taken from the PubMed and some from the Science Direct. After going from this process, you would be able to identify the articles. Now, there are type of the research articles. You can see original research articles, there are review articles, short communication, letter to editor, supplementary articles, and etc. Okay, and then we have another group, the gray area, you can say, where the books, lesser writings, pharmacopoeia's website, Wikipedia, short reports have been addressed. Okay, uh, yeah. So understanding the pathway of the research, ideally, this is what I have been coming up, and this is how my mind has been trained, and I'm able to understand that physiochemical analysis are now needs to be researched a lot because due to the solvent issues, as addressed in the previous questions, presence of materialistic quantum dots in ultra high dilutions that comes up number one. Then number two, the NMR studies differentiating structural bonds of atoms in ultra high dilutions. Okay, so these are the key questions, and these questions has been answered by various research groups. Okay, so first comes up with the analysis of the sample when you are having any homeopathic dilutions in your hand. So you should be able to trace it, whether the sample is just solvent based or whether it is having a potent any therapeutic efficacy. Okay. Number two comes up the in vitro and in vivo. So here, what we need to answer is the cellular models, the protein expressions in understanding mechanism of action of the serial diluted medicines. Okay, so you need to answer that if there is an effect of these homeopathic medicines, so you need to know whether such medicines can be tested in the cellular models. Okay. Number two, the replication of the studies on mammalian group of experimental models, okay, the mice, rat, hamsters, etc., for the gene toxicity, mutations, and safety for taking trial towards human consumption. So basically, in the modern medicines or even in this trials of the Western medicines, this is the data how they go up with. First, they identify the component. After identifying the component, they identify the potent inhibitors or the particular assays which has been showing a remarkable changes in a particular conditions, and the exploring it in the exploratory phases, explaining in the lab experiments, then further in the lab experiments replicating the data of the results into the animal models, identifying the non-absorbed adverse effect levels, and then bringing up the dose ranges and the product into the pilot phase of phase 1 trials. Okay, so finally, then further we can go with the 30 cases and understanding the data from the animal models and the molecular existences of the ultra dilutions with the specific cohort groups, and then designing the guidelines for the phase 2 trials. Okay, so over the time, there was a question that what we have thought was understanding the drug proving concept, taking the symptoms, bringing up the data, understanding the process of repertorization, validating it, understanding the symptom scenario of the patients, confirming it with the repertory or the database, and then coming up to the same minimum. Okay, so this is how we ideally go in the routine life. Okay, in the process of individualized therapy. Okay, but over the time, we also need to understand this way also that how are these medicines can be further be researched. Okay, yeah. Okay, so let me give you a time frame. If over the end users, many of them would be eager in understanding that how can you research such questions. Okay, so for the physiochemical analysis, the study period lies about a period of 1 month. Okay, so what previously people have done is they have researched on by the methods of high resolution transmission electron microscope. They have done the NMR studies, the field emission scanning electron microscope. They have used selected area diffraction, inductive coupled plasma atomic emission spectroscopy, the gas chromatography. They have used the HPTLCs also, the liquid chromatography and spectrophotometry, and the UV spectrophotometry. Okay, so these are currently the techniques. There are a lot. There are a lot. These are some of the highlighted with the research publications. Okay, so this is number one. Number two, after getting the data, you can go with this cellular model and study period. The study period required for cellular model studies are 3 to 4 months. Okay. So what we do in cellular model is we try to choose this perfect cell line model designed for your experiment. Methodology and materials in details you need to understand. Okay, then assays as per the cellular model. You need to understand which assays is feasible in testing for that particular cell models. Gene expression studies, then MTT assays and PCR evaluation to see the cytotoxic effects and safety for going ahead with the dosage, and the statistical analysis as per the experiment depends upon what variables are you going to use in the experiments. Okay, number three comes up with the animal model study, which is the period is about 7 to 8 months. Okay, so once you're getting the data of physiochemical and about cellular model, you can combine it and then you can go ahead in understanding the process in vivo in the live rats. Okay, choosing the animal model design. This need to be answered because there are different models which for example in a diabetic you have to go probably if you're going for a type 1 there are STZ streptozosin models, alloxin models, but if you're going on a type 2 there are Zucker rat models. Even the knockout mice has been used, high fat fructose diet models have been used also. So you need to understand as per your priority questions which model to be assessed. Okay, methodology and material details need to be understood similarly as those which were understanding for the in vitro also. You must have an institutional and animal ethical approval. Okay, so and approval guidelines of CPCSEA. most of the publications over the time, uh, even the journals are recommending that they need an ethical approval number of the publication. So definitely, if you have an ethical approval, the process of scrutinizing the data becomes much easier, and the outcomes which have been assessed can be easily acceptable. Okay. Then histological studies also need to be understand as per to see whether there is any toxic effects seen in the live models. Okay. And then coming up the statistical answers, again depending on the variables. After gathering this preclinical data, finally we are coming towards the clinical trials. So the parameters important for the clinical trials: number one, the institutional ethical committee approval and the clinical trial registration. Specifically, when you are working in a scenario of India, CTRA registration is a must, which gives us the details of the trials. Okay, and who is conducting it? Where are the trial sites? Whether the trial is in a recruitment phase or it is terminated, or whether it has been in an initial phase. Okay, then study design matters. Are you going with the parallel group RCTs? Are you particularly going with crossover study designs? Observational studies, are you going with any cross-section studies? So that matters a lot. Inclusion and exclusion criteria, depending again upon the research question you're going to address. ICF is one of the important thing. The ICF concepts can be initially can be brought up from the Nuremberg Code, the Helsinki Declaration, and over the time how it has been coming up. Some two important aspects in the clinical trial I would like to add is dose escalation study. So dose escalation study in homeopathy has not yet been studied and has not yet been researched. So such topic can be taken in initiative. We can go with 3+3 clinical designs. Okay, understanding the no observed adverse effect level of the safety initially, and then tapering or increasing the dosage as per the biochemical assessments. Okay, the biomarker assessment. And at last we come up with the adverse drug events. Okay, so definitely to assess whether these medicines have any effect or not. So here comes up the pharmacovigilance part. Okay, so this is a roadmap for all of humans. I've just given an idea and insight that how you can go ahead, how you can proceed with the R&D sections. Okay, yeah. So for an example, what do we know when we are in a stage of academics? What do? We try to go on a database. We go in a particular repertory. Okay, for example, if I want to have a medicine of a diabetes mellitus, what I will do is I will just go on the repertory, the shreyons. Then I will go for identifying the rubric. I will get the remedies. These are the group of remedies. Then I will analyze the symptomatology, and then I will go in understanding the level of evidence and the level of hierarchy of the gradations. And then I will come up with a particular remedy. Okay, and then I need to know that which potency would be suitable, or with the availability of the medicines in understanding or in the prescribing format. But same case in the example of diabetes, you can go with another data set. The previous slide explained you that how to extract the data is there are databases from PubMed, Sans Direct, Scopus. This is an example. Okay, so once you're working on a repertorial data, you're going to get data of this sort. Okay, but once you're working with the publication data, you're going to get ideally this one. Okay, so how we can search it out? This is a forecast data of anti-diabetic studies in vivo clinical trials of homeopathic medicines. Individualized homeopathic treatment, number one. Number two, retrospective cohort study type 2. Observational study type 2, multicentric study type 2, high fat and high fructose ceftriaxone type 2 models. Ceftriaxone induced neuropathy and carbohydrate and lipid metabolism induced type 2. Okay, so these are the published data which have been brought into a lot of research and a lot of literature and an evidence for the homeopathic. Okay, so whenever you go on a Google or even whenever you go in a PubMed, uh, it is like I will just show you. Just let me help you in screening it. 1 minute. Okay, it's not happening
Want to
here.
start the screen share?
Yeah. Start a screen share.
Yeah.
So
Yeah.
why are these things so important? Because once you have a strong database, you can stand your science can stand with a lot of examples. Rashmika, I'm not able to share the screen. I'm not able to share the screen on my side. Okay.
There's an option of screen share in the menu bar in the
Yeah,
left
yeah,
side of
yeah.
it.
Yeah, I've came up and here there is a share and cancel. I'm not able to share it.
Share.
It's not able. Okay. Anyways, let me go in the further slides. Okay. Meanwhile, I will go with the further slides. Okay. So, which homeopathic medicines were initially been used in the diabetes mellitus trials? Okay. Not only in trials, but in vitro experiments, in vivo experiments, and has been used as an anti-diabetic agent. So, Cephalento indica is more research, in the potencies of mother tincture 6030C 200C, Cesium gimbulinum in mother tincture 6030C, Whitea somnifera, Lycopodium, sulfur, phosphorus, Calcarea carbonicum, Ignitia amara, Medurinum, Thujox intalis, Nux formica, Natrum mur, and Carcinosin. Okay. So, this data of the medicines has been extracted from the previously published data. Okay, yeah. So yes. So why do we need to understand this EBM concept? Okay. So with the previous slides, if you try to understand the hierarchy of the level of evidences, animals and laboratory experiments, okay, studies, these are not involved basically in the EBM criteria. Okay, but these medicines do have some answers for the questions that whether is there any toxic effects when you go with the administration of homeopathic medicines. Okay, and possibly with my data set, what I have been researched and what I have been gathered after administering the homeopathic medicines in the animal models, you won't be getting any cell toxicity or any histological toxicity in the diagrams. Okay, yeah. So further, the hierarchy goes up. So case reports and case series are level five. Narrative reviews, experts' opinion, editorials, where there is no any design. Four and five comes under it. Level three evidence are the case control studies, which highlights the retrospective. Subjects have the outcome of interest, looking for the risk factors. Then we have level two is the cohort studies. The prognostic, the cohort has been expressed in the risk and observed for outcome of the interest. Then we come up with the RCTs, prospectives and the test treatments. And at the level one, we have the meta-analysis. Okay, so this is taken from the 2016 Modified Evidence Pyramid. Below is the link, and you can just go ahead and just check it out. So in this way, henceforth, we would be understanding the level of evidence. Okay, basically, when you speak of materia medica and all, these are the expert opinions. Okay, but when you speak of the clinical trials, this comes up in the level two evidences, and meta-analysis stands for the level one. Okay, in further slides also we will be able to understand that whether meta-analysis is feasible in testing homeopathic medicines or not. Let's see. Okay, yeah. So fundamental research and question and evidences. Okay, so you need to understand whenever you're working on a particular sample or a particular IMP, that's investigation medicine product, you need to address the questions like how to differentiate homeopathic medicines among themselves as it has ethanol as a solvent base. Okay, number one. Uh, so I think so we can have the copy links in the attendees column or in the share for the question number one. Okay, so for those who are attending, can get particularly the links over here, and just you can go and check the research papers out there. Number two is, do succussions have an impact as it compared to unsuccused investigational products in laboratory experiments? Okay, so is it compulsory that we have to succus and then we have to take the medicine? So there is a paper of arsenic album which has been studied on the arsenic induced trioxide on the mice models. Okay, so it has an impact or not. Okay, and again the law of similars has been similarly been addressed, so link can be shared here itself again. The third question is it possible to investigate law of similars in experimental models? So that is again a question that cardinal principles understanding as per the academics is a different part, but when you are trying to implement in the R&D, you need to understand. So many of them how have the researches, as we all know that alloxin is the unchilling agent. Okay, and when it is induced, it produces the condition like a diabetic conditions. Okay, but when the same alloxin, when it is used in dilute form, uh, It acts as an anti-diabetic agent. Okay, so there is a publication. Again, similarly, the previous example, as I have mentioned you about the arsenic album. Okay, so arsenic album, when arsenic trioxide has been induced in the mice, it produces the toxicity, but the same toxicity is reduced when the same arsenic album is given in a diluted form. So over the time, you need to do some preclinical research in identifying and in explaining that how law of similars is can be expressed and can be brought out in scientific evidences. Okay, then interpretation statistical test and the impact. You need to know how many variables are you going to use in your fundamental research. The groups, definitely there will be a control group, and other one will be your experimental product. So you need to confirm it that how many groups, and based on that, your statistical tests get varies. Okay, there can be particularly only ANOVA, there can be an COA. If there are multiple variables with a lot of outcomes. Okay, so you need to understand this thing. Okay, yeah. Coming to the preclinical research question and evidence. Now, this diagram explains us, even when we were in first year of the homeopathic pharmacy, that how the process of potentization is worked up. So the tinctures, we take one part of tincture and adding a 99 parts of alcohol, giving a downward strokes, then making it, then taking another part of 1 minute from the 1C, and then adding a 99 parts of ethanol again for 2C. Again, we are giving a 10 downward strokes, and this is how it goes up. Okay, so this is one of the reference link from where the images has been taken. Probably would be from the Viron Pharmaceuticals. Yeah, so and this is from ideally some of the raw data of access denied, defined as a decimal scale and C as a decimal scale and the ratio. So you need to understand that the 1x having what is the ratio. Ideally, there is the booming question that these medicines are above the Avogadros. So whether it is feasible in testing these medicines, because the chances of getting the presence of particle are very negligible, or you can say it is a more challenging part. But there have been research papers which has been answered these questions. Okay, but still it is on a debatable part whether the nanoparticles can play a role or not. There has been one paper of argentum metallicum, argentum from silver nanoparticles, published by the IIT group of peoples in identifying the harmonic effect in the lower dilution. So you can get these papers out into the PubMed, and where these people are in on the lab experiment, they have explained it that the particular dilution has some small quantity of materials present in it, which is showing the effect, the dose response effect, the curve uh, of the harmonic curve of it. Okay, yeah. So whenever you're addressing for the in vitro experiments, the questions you need to understand is which ethanol concentration is to be identified as a cytotoxic and how to study the dose range in cell lines. Okay. Basically, when you are going with this in vitro experiments, number one, the question comes up is which cell line are you going to research? Number two, depends upon the cell line, which assays are you going to be used and what are your outcomes in assessing it. If you want to see any apoptotic or any antimetastatic, so you need to understand the cancer cell lines which can be used. One minute, the slides have been changed. Yeah, which can be used. Okay. So even in the chat box, you can get the references for this particular questions, and in the dose range in the cell line. So that is a really tricky question because what dosage are you going to set when you're going for a cell line experiments? Okay, ideally it comes under the mule. So are you going to go for the 2 mule, or you can? There is a percentage of setting it. Okay, so that is again a big question. That is it. How to understand this is the okay, I will go slow it. Okay, I will go slow it. So it need to be understood these questions. Okay, then number two is how to differentiate ethanol activity with homeopathic medicine activity. As homeopathic medicines has the solvent base in it, so you need to understand whether the cells which are getting which are getting arrested, or which are getting killed, or which are not get mutated. So whether it is due to the ethanol or the solvent which is present below. Ethanol is known to have the apoptotic activity, and you need to confirm it with the medicinal activity. Okay, so that is again a big question. Number three comes up how to design the experimental studies in intro, avoiding the experimental biases. Okay, so whenever you are designing even the publications in cell line studies are a bit of challenging, because many of them, they go with the MTTSA and they try to publish it. Okay, so we are not able to understand what does the a paper comes towards with in conclusion. Okay, so you need to understand the outcomes also with the particular topic when you are working on it. And are in vitro studies sufficient to provide evidence for homeopathy? Okay, so is it necessary to go with in vitro? Because what we have thought is the process of drug proving, and that is how the data has been collected, and that is what we are learning and we are prescribing for the patients which are in the coming to us. But these in vitro data, they will answer the questions like whether is there any scientific evidence or any physical entities which are showing any activities in the cellular changes, cellular responses. If you try to understand the previous slides of in vitro, where the cell models and has been expressed, so these questions will be addressed if we go up in going experimenting with the in vitro studies. Okay, and definitely the part of statistical test and impact comes up into here. So again, it depends which variables and which other groups. So definitely you will require one control group with phenol, and then other with particularly with the medicinal group, and then you need to analyze as per the outcomes. Okay, and as per the variables, how many are you going to use it? Yeah. So again, now when you want to work in a field of in vivo experiments, that is the animal model studies. Number one, as answered with on the previous slides, approval of institutional animal ethical committee is very important. So whenever you're looking at publication also of animal models, either at the bottom or either at above of the reference, there will be mention about the ethical approval number, animal ethical approval number, or in the material section of the research paper, you will be able to get it. There are various journals, paid journals, you can say it, which try to publish the data, and these are something misleading for the homeopathic field. Okay, so we need to have a close watch over our publications also. Okay, then how to calculate dosage for administration in animal experiments related to homeopathic medicine. You also need to understand modern medicine, how they work is they try to go with body surface area, and as per the weight they try to go with the dose conversion models. That is not so feasible in testing in homeopathy. Why? Because the dosage which has been used, it is almost 20 ml, okay, or possibly 40 ml. So these are from the research paper I've been quoting it out. Okay, there is one of the research paper from Cephalendra indica and its activity and cesium gibberellin activity as an anti-diabetic. Okay, in 2013 by Sampath sir has been an Indian researcher has been research on this topic. So where the dosage was administered was 20 ml or 40 ml. Now imagine it in the when you try to convert it into the minims, it becomes 2 or 3 minims. Okay, so how can you just make a dose conversion model based on those minims and then you can convert it for the phase 1 trial? So that is again a big question, because ultimately what we do is when we are adding particularly for a pilot phase studies, we go with standard format, that 4 globules, uh, BD or twice a day, and then we try to go up in the trials and then we try to administer it. Okay, so here the such questions need over the time need to be thought, need to be in a thinking process, that can this there be any ahead any growth, whether quantum theories can play here something a big role in understanding the dosage of pharmacology, uh, something or not. You need to interlink all this thing in entanglement, all this process. Okay, the third question is, are homeopathic medicines safe in testing effects in animal experiments? Okay, so definitely you need to understand this. There is a question that homeopathy, science has to answer. If there is effect, definitely there must be a side effect also, because we believe in the causality therapy, and where there is a cause, there is an effect. So definitely there will be some adverse events. Okay, uh, at this moment I can't say it, but if you try to understand, if you try to experiment it, if you try to go on more and more dosage in the experiment, definitely you will get some of the other histological evidences where you can see that the cells are getting inflated or not. Okay, But due to the process of potentization, many of the researchers has claimed that the dilution process is itself makes it toxicity limit over the part. Okay, so there is no any presence of toxicity because the dilution has reached at so level. So the question converts not on the basis of toxicity. The question converts on the basis of efficacy now. Getting it? So even though if you're having the particles, but you need to understand whether there will be any chance of any effect or not. Okay, and to get more understanding about this, the reference links have been pasted on the chat box. Even you can go on PubMed or you can go on Google Scholar and you can type animal model research and evidences. There are a lot of reviews have been written. Even Dr. Anisul Kodabak sir, he has given a lot of review on the experiment models. Okay, we have the Paulo Beloved sir who has also given his impact. There is a group of researchers from Brazil. They have worked on it in bringing up the evidence. We have Stefan Bongmadet, sir. We have the Switzerland group. So these people are trying to bring up the evidences and how over the time the medicines can be tested and can be brought up. Okay, yeah. So and again, is it possible to identify mechanism of action of homeopathic medicines in vivo? So definitely, another question comes up that how do these medicines act? Okay, so there are almost 16 or 17 theories. Number one, we have water memory theory, can make the answer, silica hypothesis. We have harmonic actions. Then again, there is covalent bond theories. We have there are evidence of the EPR entanglement model. We have quasi-quantum models. So everyone, as per their own field, they have tried to bring some of the theoretical approaches and how it can be brought up into the mechanism. Okay, even there is one paper about the gene expression studies that homeopathic medicines can be studied, can be understood with the gene experiments. The effect of the medicines has on the gene expressions. Okay, so there is a good review on it. So ultimately, we need to know that which medicines need to be addressed at a particular time, and definitely, if for example, when I speak of diabetes, so in front of me, I have 8 remedies coming up: Agroma agosta, Syphilinta indica, Syzygium gymbolinum. Then we have acid phos. Okay, uranium nitricum. Okay, insulinum pancreatinum coming this. But based on the utilization, these remedies can be prescribed, and also depends upon the physician and his own thought process of prescribing the own individual parameter. Okay, but for those who are working on the non-individual parameters, the patent practices, definitely we need to understand that particularly which combinations can be used. And there has been a research where whether you go with individualized therapy, whether you go with non-individualized therapy, both of the therapies have the efficacy. Okay, so we have the database. Even those references can be put in the chat box. Okay, so you can go ahead. So these questions also need to be understood over the time. Okay, and again the question of statistical test and impact. So whether such data uh, is suitable in bringing up the statistical evidences. So again, the variables, the group of testing the animal models, you need to understand the control. Definitely you need to bring up. See, if you will understand the animal studies, what you have to do is you have to carry out all the animal research papers, and then you have to compare it with one another. Okay, then how many, what was the methodologies used, what was the materials used, what was the dosage. So the dosage of each experiment you will get a slight dose variations. Okay, so that you need to extract it. There is a good paper of ours published in the IGHDR, the Syphilinta indica as an anti-diabetic agent. You can go on Google and you can just check it out, and where we have given a brief understanding of Syphilinta indica as per the materiamic aspect and how it has been in ABM. In further slides, we will be going with some examples also. Okay, so yes. Coming towards the case studies, research question and evidences. Now, specifically for those who are working in a practitioner's perspective, the home case guidelines have really brought a lot of internal validity and assessment, specifically of the case reportings. Okay, so there is a link you can go here. You can even go on Google Scholar and PubMed, and again you can search out and you can try to identify the data. Okay, here you will be able to understand if you are treating any cases, if you're getting the results out there, then how you have to document it. Okay, now many of them I have seen many research papers. what happens is investigation reports have not been given in the result section. Okay, and it is said that the particular remedy has the effect. Okay, but has the effect, you need to support some evidences. So not only investigational report, you also need to see whether the information of the case were documented correctly or not. So again, we can have different sessions ahead, but this is one of the guidelines you need to understand and where you need to go over it. Okay. This I think so. Robert Wise Hansley sir has been reporting these guidelines, and it has giving a lot of importance about how you can document it, the clinical findings, timeline, diagnostic assessment. Okay, uh, again, the therapeutic interventions, whether it was individualized homeopathic medicine, whether it was non-individualized homeopathic medicines, whether it was the prescription pattern was a sugar of globules. The globules were prescribed, the powder was prescribed, the dilution was prescribed, the route of administration, whether it was oral. Basically, majorly it is oral, but many of them, there are very few practitioners who go with the nasal route also, understanding even the topical ointments, how it was used. Okay, so that also need to be entered in this home case guidelines. Follow up and outcome assessment. So definitely you need to know how you're going to assess the follow up. Many of them, I had many various questions, like for example, how to decide that which potency can be selected over the time period. Okay, so then how to decide whether the which remedy to be selected. So we have the pre-thought guidelines over here for the follow-up assessment. You can go on the website and you can just check it out with the pre-thought follow-up assessment criteria. There are modified Narinjo criteria which is used in assessing. Okay, so and definitely the quality of life also need to be assessed when you're assessing for the follow-up, particularly in the case reporting trials. Okay, and the discussion section comes up, then how the personal discussion of that particular author and his contribution, how he has been kept up. Then comes the patient's perspective, and finally with the informed consent, whether the informed consent was taken up, whether the patient was introduced about how are this, what is this homeopathic system, how it is practiced, and what are the chances of it. Okay. Currently, in this pandemic, various people are claiming that these medicines can be used, this can be used, but you need to identify first the whether the particular trials have been registered or not. Okay, so we will go ahead. This is one of the important aspects when you're working on clinical trial research question and evidence. These questions needs to be addressed. Okay, Rashmika, can we have a screen sharing option here?
Just try to screen sharing. Try to share it now.
Yeah, just I will just check it up.
Yeah,
Opening browser. No, it's not happening. It's not happening.
it's not. I've given you the permission to. I've given the this
Okay,
thing.
let me. Okay, in upcoming sessions we will try to do
Yes.
it up. Okay, okay. So whenever you're working in clinical trial research, definitely trial registration is an important part. So you must know that whether the trial was registered in Clinical Trial Registration of India or not. Okay, so CTR registration is there or not. So you can go on Google, you can go on CTRI, you can go on that website, you can run a trial search, and there you can type on a search engine based homeopathy word, and then you will get all the trials currently running in India. Okay, of particularly of homeopathy. There are publications which do not have mentioned the CTRI registration number. So such publications might be a little bit of uh, weak evidences. Okay, and the validity of internal and external can be questioned. Okay. Similarly, there are a lot of trial data. For example, we have ENIH, we have clinical trials.gov of American trial registration. So you can go there also. GCP guidelines should be followed. Approval of the IAC committee to be understand. Type of study designed to be used in trial should be mentioned. Then are the homeopathic medicines safe in administering or not? In the chat box, you can go on the Equator network for clinical trials. You can just go and you can just see it. There are particular guidelines. So there's consort guidelines, Prisma, okay, which can be used. And again, the part of interpretation of statistical test and impact should be understand. Okay. Clinical trial also need to be understand when you're working on homeopathy. What is your objective? That also need to be understand. Your primary outcome, your secondary outcome. Okay. So basically, what we need to answer is the placebo trials, because 2005, the data was more impacted on the basis of whether it is a placebo effect or not. So definitely, as the solvent issue is there, I hope so you would be getting from previous slides. You need to have one of the comparator group of having the solvent or an ethanol, and another you need to have homeopathic medicines, and the method of administration, that is also important. Whether you're using an individualized, you're using a non-individualized, you need to understand these things also. Okay, coming to the systematic review, research, question, and evidences. There has been a lot of data over here. There is a group of meta-analysis group. The Cochrane database currently uses all the about the meta-analysis of individualized and non-individualized homeopathic practice and its outcome need to be assessed. Modified validation of randomized controlled trial need to use when looking effects of homeopathic in particular trials. There is one good research paper from Dr. George Mitchell Kosser, the serious mistakes in homeopathic trials. You can address that, and then you can understand how this trial need to be administered. Difference between meta-analysis studies of modern medicine and homeopathic medicine. So definitely you need to understand how are the Western medicines meta-analysis bringing in evidence in the market, and how are this homeopathic medicine bringing evidence in the market. Then for this you need to definitely have a Prisma chart and effect models, fixed or random, and question of homogeneous or heterogeneous data in homeopathy. Okay. So again, there is the link of meta-analysis studies. Even a lot of people have a question that how to study these say meta-analysis studies, and are this meta-analysis really helpful of bringing the evidences. So for this we also need to understand how are these studies been conducted. Okay, are the risk and bias factor considered while taking the clinical trials? Then were particularly any particular how was the outcome assessed, based on heterogeneity or based on the homogeneity? Okay, the effect size, how it was expressed, the forest plot and the funnel plot examples also need to be evaluated over the time. Okay, now coming towards the drug profile comparing with Medical research outcomes. Okay, now from past 1 hour we were just trying to understand the research questions, probably how you can come up a thought process, and then we were having particular copy links pasted in the chat box. Okay, now how we can bring a particular data. So for this means the you can say in a nutshell, after studying this what are we going to understand? Okay, so we are going to understand the perspective of homeopathic mathematica, pharmacopoeia, basic fundamental experiment, cell and experiment, animal model experiment, case reporting, clinical trial. For example, if you're studying Calcarea carb, now how we were taught in our academics, Calcarea carb, the fair fat flebby, the pictorials, the modalities, the remedy relations, the cravings, the aversions of the Calcarea, the rubric identification. Okay, so if you try to understand the parameters evaluated as per the empirical sources, drug proving is one of the parameters to assess, means to understand the Calcarea carb. Okay, it is one of the qualitative method. There are case studies mentioned by various pioneers in their lesser writings. Okay, even in their particularly when you study a Kent's Materia Medica, Kent has expressed a lot of descriptive data, of understanding the Calcarea carb. Okay, so you need to understand this. Ideally, these are the level 5 evidences, as we previously has been understood the pictorial diagram of the level of evidences. Rubric again is a qualitative mode of selection. Your Bayesian approach again needs to be interlinked. Remedy selection again is a qualitative mode. Homeopathic Materia Medica again it is a descriptive data. And Pharmacopoeia finally in HPI volume 1 you will get the data of Calcarea carb. But this is one of the phases, one of the brains when you are in academic. Okay, if you wanted information about Calcarea carb, you can take it. You will be thinking on this perspective. But over the time, now if you have understood the previous questions of in vitro, in vivo, the clinical trials and the meta-analysis, then you will be able to understand or physicochemical analysis, the parameters evaluated. So now spectral analysis based on the ABM, you will be able to understand is there any OH group change in the Calcarea carb of 6C and 30C potency. In vitro cancer cell models, anti-metastasis activity is seen in Calcarea carb. Okay, so I think so you can paste the links, Rashmika, on the chat box. So again, in vivo murine melanin model of skin cancer induced direct dormancy and malignancy. Okay, so in vivo chicken experiments, the healthy quality of battered chicken improved when after prescribing the Calcarea carb. In vivo ostriches, the thickness of the eggshell again improved it, and case reports of fibromyalgia, infertility, obesity. There were clinical trials also about the Calcarea carp. Okay, so whenever now you're understanding a particular remedy, example in Calcarea carp, we must now have a broad sense of mind. Okay, we shouldn't just focus on the provings and along the lesser writings and along the stalwarts what they have been studying, even the repertory. We must have an add-on thing. Okay, even the pharmacopoeias need to be get upgraded. Whenever you go in the pharmacopoeia, we have particularly a section of Calcarea carp. It's physical entities, test of solubility present, then inferences, then proving who has been conducted yet. But when there are currently a lot of researches coming, you need to understand this data also, and which will again expand the scope of R&D, and even definitely will expand the in understanding of MO mechanism of action of the homeopathic medicines. Okay. Yeah. So coming up on the end slides, I would like to give you one of the most current affairs and events in India. What is happening? If you try to understand, there are total government colleges are 32, private colleges are 185. Among this, yearly 15,000 approx undergraduate candidates are getting passed out from the academics. Okay, and from PGs there are over 1,000 candidates who are getting passed out. We have around Organon of Medicine 242 seats. Okay, Repertory 229, Materia Medica 248, Homeopathic Pharmacy 75, Practice of Medicine 136, and Pediatrics 101. So this data has been taken from the CCH 2018 college list. Okay, and the grants given for the admission process. Okay, and here you can see the roadmap that which state has the maximum colleges. So Maharashtra is in the leading phase as the production unit of the graduates. Then we have second is the Gujarat, then we Madhya Pradesh, and this and this you can see it. Okay. The question is, when you are having 15,000 brains coming out in that particular year, so we all need to retrospect our own selves. Also, we need to introspect our own selves. That are we potent enough to face the challenges, particularly when you're going out? See, you when you're going to get a degree. Okay, so it is not going to matter. Okay, what is going to matter is when in a community, in a scientific community, are you able to explain the people what research have been done? Okay, so if there are 15,000 minds coming out every year, if each of them or a group of them try to address the previous questions and the databases, and if they try to understand the databases, and if they try to put forth this questions into a scientific evidence, definitely we will have a lot of impact on the global scenario. If you try to understand the previous slides, getting it, the impact on the market size, the impact on particularly IMP products, getting it. Yeah, so the important point coming up is the SWOT analysis. 2017 was the year when the slides were prepared in my academic times. So I thought of bringing it this. There were two or more research groups involved in this. So strength, weaknesses, opportunities, and threats. So you must know what are your strengths. So currently, homeopathic philosophy, Central Council of Research in Homeopathy, and CCS. These are our strengths. Okay, when you're working in India, these are our strengths. Highly efficacious and low hazardous in extremely dilutions, cost-effective treatment and cost-beneficial natural's law. It is potentials for alternative health justice, stability of homeopathic medicines in high dilutions. Okay, so you need to understand. And weaknesses are the emergent cases, publication bias, surgical procedures, diagnostic protocols with the treatment guidelines, validation of standard operating procedures, intellectual validity with the results, follow-up assessment, scale validation with the respect to particular remedies. Okay, you need to understand. Opportunities are the MOUs. What is the apex body of CHR which is doing MOUs with the other countries? So that is one of the most strongest point for us. The rural health attraction of the particular programs which are running. Even today, we have the NRHM programs which are running all over the states. So these are the opportunities where we can elaborate our scope. Then we need to inbuild more government colleges over the states, so that we can have a lot of funding with the particular projects, as the private sector has a lot of impact and a lot of pressure on their admission process and about their conducting this examinations scenarios and all, with the inspection scenarios and all. So we need to have well established colleges with like one, we have a reputation of NIH, we have NEIH also in Eastern. So such colleges need to be get broader and broader. We have the particular government, particular state has its own governmental colleges. Okay. This R&D section of the policy need to be reframed in the academic as well as in the private sector also, what all things to be done. Definitely pharmaceutical companies need to get R&D section elaborated. People who are performing with the postgraduates also need to be pulled up in this pharmaceutical companies so that the R&D section can get strong enough. Biomarker analysis, experimental studies need to be studied. Skill development courses and extracurricular need to be understood. What are the threats? As earlier, we have gone with the Lancet publication. So media and the way it is propagating is one of the most important threats. Okay, there are Drug and Magical Remedy Act. Tomorrow, I can't go ahead in the crowd and I can say that yes, homeopathy is effective in such and such conditions. I must have some of the database. So what have we learned is how we can generate the database, how you can bring it, and how you can bring the data from level 5 to level 1. Getting it? So this definitely is impact. Now Australian reports of NRHM, it has been you can understand how it has made selected studies, the data was brought into it and the outcome was disclosed. So such things need to be brought is one of the threats. Okay, then NMC bill, even we have the Swiss reports and medical legal cases, again with the high court cases going up. Okay, even the administration process needs to be very highly meticulously understand and to be supervised. Okay, so what are the further research questions you can address? Are the pharmacoeconomic studies, number one? Number two, pharmacovigilance studies, mechanism of action, and their comparative analysis with established mechanisms. You need to replicate the same mechanism. Is it possible to replicate or not? Comparing vaccine trial with homeopathic trials, crossover study designs, dose tapering models in trials. These are some of the new things which we need to do. A KPA model is running from ancient times. Okay, not sorry, from the empirical sources. So you need to understand first. You need to bring a lot of work on this and meta-analysis studies. Okay, so this was all about the session. Rashma, can I have you on my screen?
Yes.
Okay, so these are some of the details about me, and there are a lot of things I have definitely missed it. And as discussed with the coordinators, there are a lot of things and a lot of in-depth analysis,
True.
lot of research papers. So within a session like this, I have been just
It's
trying
very
to give my
difficult.
best, and in identifying. Okay, uh, later we can go with some more sessions, how
Definitely.
to go for a database, how to screen it out, how to remove papers, how to segregate it, how to drag down the information, and how to build up your own materia medica. Okay, because materia medica is based on the expert opinions, but every individual has its own materia medica towards its understanding. Okay, there are teachers who try to understand as per their knowledge and as per their references toward us, but we ourselves need to get elaborated and need to expand. Okay, so this is what I think. So the project gets to
Thank
over
you
here.
so much. Thank you so much,
So
Aditya. It was a wonderful seminar. Like you said, research is a vast, it's like an
yeah,
ocean of content, and we cannot, put the ocean in 1 hour to
yeah,
talk about it
exactly,
in one
exactly,
hour.
exactly,
So
exactly.
I hope we have more sessions. Now we'll start with a little question and answer session.
Yeah, sure, sure, sure.
Yeah, I'll start posting questions as well from Facebook as well as in
Okay,
this. Uh,
okay, okay.
Listeners,
Hope
if you
so.
have
Yeah, yeah. Hope
if
so.
you
The end
have
users and the students and the audience are getting it what I was speaking over it, because in the live session from past 1 hour I was just trying to hear my own voices. So let me see with the questions I would be able to understand about the feedback of the sessions also. So
So, listeners, if you have any questions, please post your questions on the comment section so that one by one we can start posting. I will on the Q&A post so that we can start posting.
okay, okay, yeah, sure, definitely. I'm ready for it.
Yes.
So
Okay, there are so many questions coming on, soaring in. Great. So, the first question was initially asked. Okay.
okay,
This was asked by, yeah, this was the first question asked.
okay.
It was asked by Sohan
Oh, this.
Shiv Chauhan.
Okay.
Shiv Chauhan. So if you can answer, why some medicines, a single dose works for 10 days, or more. Yeah, 10 days or more, but for someone it's just 2 or 3 days. If you can answer this.
Yeah, this is one of the controversial questions, and this is what it is very difficult for me right now to answer it. Okay, such questions can be addressed on the basis of remedy identifying the particular action as per the follow-up databases. So you can do one thing. You can first understand the modified Naranjo's criterias. You can understand the Prathot, and when you're going up with the case studies, what you can do is you can take the follow-up, the remedy which has been administered. You can take the date of registration of that remedy, and then with a further follow-up, you can add a quality of life scale and particular biomarker. Okay, so what you can do is particularly, for example, if you are working in a trial of diabetes, so you can go on the HbA1c levels, number one, number two, BSL fasting PP, and lipid profiles. Okay, so if you're choosing a Cephalendra indica in this perspective of administering, that again depends whether you're going for a mother tincture or you're going for a potency. You can take a data from animal model. You can assess the HbA1c levels, what had you have been observed. Okay, for example, in animal study of 28 days, you will be able to understand the control group of ethanol controlling the sugar levels, and the homeopathic medicine Cephalendra controlling the levels. Okay, you can compare it. You can keep this data, and when you're prescribing for the person for Cephalendra indica, so you can see whether the same data of 28 days is it reproducible in the clinical trials, in the pilot studies. Okay, so that might be an option, or that might be an a starting to answer this such questions.
Hey,
Okay, and another thing is definitely there might be my lacunae also, because we need to go up with a lot of databases. Okay, so answering such questions can take a bit time, but this is one of the thought process which I have expressed it. So you can think on that basis, and then you can come up, and then you would be able to understand that whether it is possible or not to test it. Such hypothesis, is it possible to test it or not?
the next question is
Yeah.
from Dr. Jay Shah.
Okay.
How to conduct ethical committee approval in small individual clinic for a clinical trial?
Probably this question, I think so. We can take another session on having an approval on ethical committee, and definitely there is there is a website. Okay, can I have a slide sharing?
Yes, 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 definitely. Yeah, if
One
you
minute.
are able to share your screen.
Yeah, actually, I'm not able to share my screen. Okay, what you can do is you can go on Google. On Google, you can go on Institutional Ethical Committee. Okay, this is the searching words. Then
I
you will, yeah. Is it possible for you to do it?
Yes, institution ethical committee.
Yeah,
I'll just
committee.
share the um,
Yeah,
link.
there is a state-wise list of institutional ethical committees. Okay, so in Maharashtra, I think so probably there are about 50 or 60 ethical institutional committees. So what you can do is you can just try to address first the nearby around your area where you want to conduct the trial. You can address your research proposals, and there you can go on it. Okay. Now, in the small institutional setting up the trials for the clinic, it is difficult for me to bring all these things right now on the table. But definitely in the coming times, you can send the mail ID. I can send you some of the important links where you can be able to understand, and where you can set up your you can have ethical approvals. How you can do it? Okay.
Great. I have posted the ethical institution of ethical committee on the
Uh,
this thing. So if you can see, you can go in for the same.
Yeah, and also I think so. It's of only of Delhi. There are every state has its own ISC recommendation, so you can just go out and you can just check it out. Okay. So
Yeah.
definitely, it is also important in academics also from which college you are studying, whether that particular college has enrolled any clinical trial in a particular registered in the CTRI. Okay, so you can also assess the internal validity of the colleges also, which college from homeopathy is been going with the clinical trials, so that there are a lot of questions during admission sessions that which college to get to be taken in the admission process. So you would be able to understand if you want to work ahead as career in R&D section, you need to know that which college is coming up with the clinical trials, which trials has been running means as answered previously, that CTRI, you can go on the website
Yes.
of CTRI, you can go on there, you can just click on it, and you will get all the clinical trials running in India of that particular database. Okay. Yeah.
Yes. Um, Karthik has posted if a remedy is in a potentized form more or
Okay.
less than Avogadro numbers. So, can we be able to show proof that homeopathy works with our present diagnostic tools?
uh, for remedies in a potentized form, more or less than Avogadro. So can we able to show? Definitely, because as previously posted the links, you can go through the links. Number one. Number two, what we can do is the roadmap, how to develop the evidence for such questions has been answered in the previous slides. So it becomes a question of which methodology tools are you going to assess. Okay, if the in a potentized form, you need to know that whether you want to assess the NMR spectral studies, whether you want to assess the H1 bonds, whether you want to assess the particular impact of the succussion. I think so. 2020, there is a latest publication from Stephen Bowman, sir, in the scientific report of Nature, that impact of succussion tested on evaporation droplet method. So you all can go through that link. You would be able to understand it, that what all methods can be used. Okay, and yes, initially, see, when you first need to have a database review. So, potentized form of medicine, you can use succussion as a keyword, and you can use homeopathy as another keyword. And another keyword, you can use a physiochemical review or an analysis. Okay, and then you can first gather the data. You can scrutinize it. What all methodologies has been previously used, and what all outcomes have they have got into their research papers, and then you can understand whether this particular tool is sufficient for testing or not. For example, the IIT Bombay research had worked on metal group. Okay, so there has been you need to understand the sources of drugs also. Basically, mother tinctures, HPTLC, LCMS are the good criteria to understand the particular alkaloid contents. Okay, and when you are going up in the potentized form, definitely you need to understand whether it's the same methodology is easy to identify or not. Okay, even you can go for HRTM analysis currently, or the droplet methods also. Inductive couple plasma methods also, you can go it on it. Even there is a DLS also. If there is a presence of particles, you can go for dynamic light scattering methods also. So ultimately, it depends which product of homeopathy are you testing for a physiochemical analysis. Number two, which potency are you testing it, and definitely risk benefit ratio, because all these things are going to require the cost and budgetary norms also. So you need to understand whether it is easy to test it or not, and the replication of the experiments. With the first reading, you cannot conclude it. You need to do the experiments 3 or 4 times, so that you can get an average reading, and then you can come up with it. I hope so.
I have shared the link of.
Yeah,
Okay,
yeah,
there's another question. If
yeah.
our medicine is dynamic, is a dynamic form in energy
Okay.
form, there are any diagnostic tools to detect it and observe change happen after. Yeah, I will share the other link also along with it.
Okay, okay, okay. This
Yes,
question
please,
is very
please answer.
interesting.
Yes.
And for this question, first of all, I would like, there are various of my colleagues who are currently been working in understanding this phenomena. You can say the question can be answered in the form of wave theory or wave particles. Okay. Because dynamic form itself indicates that the entities of traceable materials are of not any relevant, and now you are in a phase where the direction concept of direction doesn't applies. Now in this form, you have to go with quantum theories. There is one I think so there is one of the research paper of quantum theories of homeopathy. There are series of papers, part 1 to part 7. Okay, they haven't explained enough. These studies definitely can in coming times might be a key to answer it, but for answering such questions, you also would be needing such resources. So for such things, you have to approach for a particular institution, particular institutional body, or a physics candidate, particularly who is working on any quantum theories. Okay, so it matters a lot uh, when you try to answer this. Okay, yeah, what is this? Whether it's matters or antimatter means I'm not able to understand this. Matter or antimatter based on the.
This is the ResearchGate profile. I mean, sorry. Oh, this is the profile where he has given a. You have published a thing on understanding high dilutions, right?
Yeah, yeah, yeah.
The
Actually,
same.
it is in a publication process, a publication process. So
Yes.
it is going to come out. The off prints are available, but probably you all would be getting in coming times. Okay.
Yes. So you
So
can follow him on ResearchGate. So you can read
yeah,
his publications also.
yeah.
So
Even
that is what.
I would like to address some of my few mistakes because I was a student in my college and probably there was a desire and passion that I want to go for a publication and I want to understand how it impacts. Okay, many of my initial publications are in a paid journal. So various of my seniors and of various of my respected teachers and in the field of R&D, they had shattered me a lot. You know, they had asked me so questions and so much of defenses that that has brought up me in enlightening this thing. So the question of publication is really an important part because if you're taking R&D as a career, that definitely you need to know that your publication must be a peer reviewed. It shouldn't be a paid. Okay. So these mistakes I have made it. You can go into my profile, you can just check it. But I don't want that the further young budding homeopath should do it. Okay. So you need to understand first the database and the literature, and then you can go ahead and you can address it so that we can have further a good international platform and we can have a good discussions among us in understanding the concepts. Okay, so this is what I would to like to give my personal messages to all of them who are
There is
in that position.
another question by Vinay. Can we
Yep.
compare the susceptibility with the gene expression in different cell of the body and then form a good organs for particularly their functions?
Totally. Right now, I can't answer this question because it is part of R&D, and definitely it is an interesting thing. But in coming times, we can do one thing: collaborating the research with a particular R&D groups. Such questions can be twisted out. But sorry, right now I can't answer it because it requires. I need to go with the particular understanding of this question, and then I can answer it. Yep.
There's another question when you said about
Okay, when you
wave
said about
theory.
wave theory, is there also involvement of matter and antimatter theory with homeopathy? Depends again if this question can be diplomatically answered right now. Number one, if there is a matter, definitely there has to be an effect. If there is no matter, then definitely it comes into the wave theory. So in homeopathy, it is about the analysis and how the R&D is working on. Because if you try to understand, there are some group of researchers who are understanding homeopathy based on nanoparticles. There are some group of researchers who are understanding homeopathy on base of wave theory. So I think so. This is the third question coming up with the same context. So again, I need to make my own database and then I can answer it all. Right now, I can't be able to communicate it.
Yeah, that's absolutely
Okay.
fine, Aditya.
Yeah.
Let's talk about the basics. Like if a
Yeah.
person who is now an undergraduate, like you
Yep.
said, when you were in your undergraduation, when you
Yep.
were
Yep.
in your
Yep. Yep.
college, you had faced so many problems and so
Yeah.
many
Yeah.
issues.
Yes. Yes. Definitely.
Starting from choosing a career path after finishing PHMS and after wanting to do something more on it or wanting to explore on a scientific base, what was the difficulties you had faced and how did you overpower it and how did you cross the whole bridge and time?
The In a nutshell, if I want to answer this question, this is very interesting. Definitely, number one, the thing matters are the resource person. So you need to understand which resource person are you communicating it and whether that particular resource person is valid in answering all your questions. Okay, number one. Okay, number two, first you must need to understand the scope of taking this science. Okay, so many of them has a question that if we take homeopathy in the field of undergraduate, then what are the further scope in the career? Are we able to economically stabilize ourselves? Are we able to gain all our introspective questions, or and are we able to stand in front of it? So my answer for this question is definitely no. Any field is weak in itself. It depends upon your impact of knowledge, your impact of passion, and your dedication. Okay.
Very
In India, as I explained you in the previous slides, 15,000 brains are coming out. But the way the one are approaching in the R&D are very few.
very
So again, it depends. Each question depends upon what resources and what persons are you communicating it. There are some questions that I today even might not be able to answer it, but even today I try to understand the research papers and I try to communicate with my subordinates or my companions. Even I communicate with my college teachers also that is it feasible in understanding this paper or not, and how to understand this. There are various clinical courses. There are various online courses you can go towards at the session. Like there is EDX, there is Red Hoop, there is Coursera, which performs the distinct learning courses. Okay. Number three is what are the scope of homeopathy once you are getting out from the graduation. Okay, so you can either join the governmental sector of the jobs, that is NRHM post. The Indian government has initiated a lot. You can go for state government. Right now, for the COVID situation, there are a lot of Ayush medical officer posts coming onto it. So uh, depending upon the impact of the depending upon the demand of the market. Okay, so again that again demands. Okay, in R&D I can say you, if you're having a bit of capital, then you can stand good in R&D, because R&D requires a lot of capital coming into it. Discussing the things and the time is a very important part, because if you want to really work in the experiments, you are going to invest your time into it. So
yes,
the budgetary session, the time, and the resources what you're going to use, that needs to be again assessed. Okay. Another thing are the academic teachers teachings. Okay, so definitely this is my sincere request for, if there are viewers who are from the academic background, that we means the pattern of education need to be changed. Okay, what are we teaching were the initial, as you can see the slide of Calcaria car. So how we were taught, and now we need to implement these things ahead also in the slides also. And even if there are such publications, then definitely the undergraduate must have at least an acknowledgement that these are some of the publications which are getting highlighted over the time. Because if particular research group is taking so much of interest and investing their time, their budgetary sessions, and their intellectual IP rights into it, then why aren't we acknowledging it? Okay, why only the few research group is trying to acknowledge money, getting it? So imagine if we all around, if we are all well versed with this type of studies, that definitely the upcoming times, that is 2025, would be the time where we all will be having a webinar in a very scientific basis, getting it?
yes,
And definitely we will be sharing our own publication details. Right now there are people who are sharing the details out, but if you categorize it, there are very few and very rare, and those are the expert brains, I can say it, because they have taken a lot of pains in bringing up the evidence.
yes, very true.
Yeah.
Like the slide you shared, I think everyone has to see the slide. The SWOT analysis.
Yeah, definitely
Uh,
the SWOT
I
analysis
hope the opportunity and the strength column increases and the weakness and the threatened column decreases.
decreases. Exactly,
That's all
exactly,
the
that is the ultimate
focus
aim.
here.
Because we want to see, we have to be optimistic, but same way we don't have to be a skeptic, getting it?
Mm.
Because
Yes,
in the whenever you go outside, people are like, okay, homeopathy, okay, this seems to be a different science.
yes,
There is a recognition, this science definitely has a recognition, but it depends on us also, and in individual, and and with everyone, that how is anyone taking this science into it, how are they interpreting it. So definitely sometimes there are failures. Even in my experiences, I would like to share that I was conducting an experiment. I had 11 rejections for that particular sample outcomes. Okay, but
okay,
I didn't stop. My experiment didn't stop yet, and currently now it is going to come up with the publication. So that is something I'm going to. Once it is in the publication, I will definitely share all the link. Everyone, you can even follow me. So you can imagine. So there are failures. Always you are not going to get the success rate. Getting it? Each research paper will teach you the way of the experiment has been designed, what all thought process has been involved, and definitely the best thing is the you have to approach the particular person who has worked on that research paper. That is the best thing. The one who has published, you have to contact directly to that particular. You cannot directly to that particular person. Okay, do not assume each and everything. Try to question it, that why such things is happening, why are we studying it today, Okay, and what is the impact going to be ahead in the life, and is it significant in studying such things? So we need to also retrospect ourselves over the time. Are we able to analyze ourselves? Are we able to question? Even there are a lot of debates. Okay, so such database and such literature can stand you ahead in the debate also, and can bring you a lot of differences.
Yes,
Yeah.
very true. Uh, one again interesting question from Dr. Jayasha
Okay.
was about dark energy being related to homeopathy.
Yeah. So again, right now, probably there are the same. There was a the same in this. What I want to say. Actually, I'm not able to recollect the particular article. I will just
Okay,
post you once I get into it. So
definitely.
definitely, uh, these questions again, as I told you, need to be answered on the basis of the expert opinions. Okay. Now, as per my analysis and all, right now I cannot answer such questions. But you need to read it a lot, and after reading it, you need to understand whether you have understand it or not. Okay, there are people who try to read some two articles, then they try to put forth some of the other hypotheses, which has not ideally been justified. Okay, so right now, I think so. You can take an email ID, and definitely we can send a lot of research links over it, because
Definitely.
answering the question might take again a more half an hour, and probably it would expand the
We
session.
have
Yeah,
already
because
crossed the 1 hour
we,
limit of our session.
yeah, definitely. But we do have the answers for it. If you want, you can share the link over there, and
Definitely.
you will get the papers of it, and even you will get the review commentary paper also. So we have the such databases. Yes.
Nice. Wonderful session, Aditya. I mean, it was a huge
Okay.
session. I like I
Yeah.
said in the start, it's a very vast session. I mean,
Yeah.
vast topic, and talking about it is a very, you can't do justice to the topic in 1 hour.
Yeah. 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 Yeah.
Like you said, like from the start of, the, collecting the data, like from the evidence-based methods till the meta-analysis. It's a I think every session, every sector or level would take at least an hour to talk about.
Yeah. Definitely. And yeah.
From the case studies or the clinical studies or the
Yeah.
randomized control trials, it would definitely take, like, especially while talking about meta-analysis, and if we're talking about it, then we need to talk about the criticism that we have faced as
Exactly.
early as
Exactly.
2005, or the current scenario which is going on. I think we will be talking like a
Yes.
lot.
Yes. Yes.
Karthik has posted one last question. Maybe we'll do that one question in a few minutes,
Yeah.
and then we'll end the session. Yeah.
Sure. 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 Okay, this is interesting. Instead of proving homeopathy, can we focus on proving vital force, like inventing a new diagnostic tool to prove vital force so we can change outcome of homeopathy? There are, yeah, Mr. Karthik, there are research papers, the gyroscope model and the quasi-quantum model, which has been answered this question, but that is a hypothetical paper. Okay, so definitely in coming times, there might be possibility, but I can't assure you right now, because I still need to understand in depth, because I think so. This was the fourth question coming again. In the paradigm of the dynamic and the wave theory. Okay,
Yes.
because again,
Yes.
vital force is something which you can sense it, but you cannot see it. Okay, and it is again, it is the more debatable. There are two or three research papers. You can go through it. You can have your own understanding, and further we can have a particular mail ID share with, and then we can
Definitely.
go ahead with this collaborations also
Definitely.
with it.
I think you've given justice while answering the questions. You were,
It depends upon the audience also, that whether they have been persuaded or not, because sometimes sessions are heavy, and it depends upon the attendees also how they are attending it. So
very true.
again, it is a two-way process. So
Definitely.
it depends upon the collaborations also now.
Definitely, definitely. I hope that there are further sessions where we can split and break down each and everything and
Definitely.
keep it in a very simpler form and talk more and have
Sure.
interesting
Yes. Yes.
conversations.
Definitely. Definitely.
Scientific bases are one, philosophy is above us. So, uh,
Exactly. There are two brains currently working in the market. One is the one of the religious belief, where you cannot break anything, and one of
break
the
anything.
scientific dogmas. So this is a conflict which goes on, and we have to be somewhere in between this.
Yes,
As to translation research is a new concept coming up in the market, so definitely you all also you can go on Google. You can first learn what are translational research and what is it all about, so you would be able to understand what past 1 hour what you were being understanding through the slides is nothing but coming up with the translational research. Okay, so I think so coming times will be a much more enthusiastic if we have a lot of crowd and having a lot of interactive
definitely.
sessions.
Definitely, definitely.
Yep.
A few things before ending the session. This
Yep.
again, just like I started my session, this was a snippet of research in homeopathy. Research is a vast thing and upcoming thing in homeopathy. So I hope every clinical practitioner, every student coming out of taking up homeopathy or coming out of undergraduation would understand the importance of research in homeopathy, would start learning what research in homeopathy and how scientific bases are important in homeopathy and at early stage, so that they have their clinical practice. Or if they want to get into research, they'll have a stronger base and they'll have stronger mindset to answer the public, because social media is everywhere these days, and everywhere people cannot be there to answer every criticism or
Exactly.
everything in
Exactly.
the world. So that's one thing. The second thing is, again, this is just a snippet, so we're planning to do a session. Those who are interested, those who have wanting to pursue research, who are interested in research or are in clinical practices and wanting to do a little more, step up their clinical practices in publishing these evidence based case studies and reports. We're planning to have a split session, maybe a 4 or 5 session series. So please contact us, and we'll work on the series, and we will put everyone together, and we will, schedule the dates and everything else. Hopefully, everything goes
Yep.
well, and we'll have more sessions on this, and we'll discuss more and interact more and debate more on these sessions.
Definitely, definitely. The debate is the most important part
Yes,
because understand if you understand that definitely you can have a lot of debate, and I
yes,
believe in
quest
debate because that brings up a lot of courage and you have to be a lot of updated.
definitely.
And this is a
Questioning
time,
is the
this is a
first
high
step.
time that each one among us need to answer it. That if anyone speaks up about homeopathy, anything, you must be scientifically sound, and then on what basis, which question to be answered. That also need to be understood.
Yeah. Likewise, questioning or raising questions and raising curiosities is the first step for going
Yeah,
to the right path or leading to the
yeah,
right road.
yeah,
So hopefully we have more sessions. Thank you so much, Mr. Karthik, for giving me such an opportunity, for
yeah.
having
Right, right.
the whole session. Giving me this platform to conduct such sessions. And please visit I Love Homeopathy page for more such webinars.
Yeah,
We
please
conduct
please
it.
subscribe also. I love homeopathy, and thank you so much, Rashmika, for after a long time I have been coming up and giving a session right now.
Yes.
So that also has means brought a more of motivation into me also, and the further R&D to be improved.
Yes, yes, very true.
Yeah,
Thank you so much, Aditya. Thank you so much.
yeah,
Let's hope we meet again. Thank
sure,
you so
sure.
much. Thank you so much for everyone for attending us today. Thank you. Have a
Yes,
great weekend and great
yeah,
week ahead.
yes, yes.